Tor. It's really my pleasure and honor to introduce him and to have him with us today. I've known Tor since he was a graduate student when he was at the University of Michigan. And I was at that time part of a MacArthur Foundation funded network on mind-body interaction. That was a highly interdisciplinary group of scientists trying to make some progress on elusive ways in which the mind and brain and body are interconnected. And actually, the work that Melissa Rosenkrantz is doing on asthma first was birthed in this network. The first study, the first randomized controlled trial we ever did of mindfulness-based stress reduction was birthed at this network. So there are a lot of interesting things that happen. And also, Tor Weger's career in many ways was deeply impacted, I think it's fair to say, by this network. So his advisor at Michigan was Ed Smith, a very famous cognitive psychologist who said, I've got this amazing student. And, you know, this was a network of senior scientists, and we didn't really have students coming to the network meetings, but Ed said that, no, Tor is very special, and we should have him part of this network. And so he really became part of the network. And everything that Ed said about Tor was true in spades. And one of the products that happened at this network was launching research on the brain mechanisms that were associated with placebo responding. And it led to, and Tor took the lead on this, and it led to a paper that was published in Science that Tor was the first author of, and it was a collaborative study that several of us contributed to in the network, and that became a very highly cited paper in the history of research on brain mechanisms and placebo responding, and I think kind of launched Tor's career. He then went to Columbia, and Ed actually was a faculty member, went from Michigan to Columbia, and Tor followed him. Tor then was appointed to the faculty at Columbia. He then moved to the University of Colorado at Boulder, where he continued his work, and Tor is now at Dartmouth. Tor has done some really seminal work and is in my pantheon of scientists who are harnessing the opportunities afforded by functional imaging in ways that are much more granular and precise. And one of the things that Tor did is use functional MRI to see if we can measure pain in a more objective way, and had a very famous paper on the neural signature of pain that was published in the New England Journal of Medicine. That really was a seminal paper and illustrated how we can use these novel imaging procedures in ways that might be clinically useful. So I'm going to stop soon, but I see that you still haven't, you got the slides? Yes, okay, great. So the other thing that I just want to say about Tor is he is one of the nicest, most collaborative scientists that I know, and he has been enormously helpful in some of the work we've done. A student in our lab came from, who was a former student who was working at Columbia, got to know Tor during his time at Columbia, ended up doing his dissertation on pain and meditation, and Tor was a collaborator on that paper, which actually came out not too long ago in the American Journal of Psychiatry. Um, uh, and uh, and so Tor has been involved in a number of things that we have been interested in doing, and you'll hear much more about it today. So let's all please welcome Tor. Thank you.
Well, thank you. It's such a pleasure to be here. What a wonderful symposium. And I hope to provide a little bit of light entertainment following the talks of the giants in the fields. But I have to say, I gave my job talk on placebo effects, got the job at Columbia before it was published in Science. So there's hope. You don't need a you don't need a science paper. All right. So today, I'm going to take a different tack. I'm going to talk about how thoughts affect feelings. And this is important for all of us because feelings directly or indirectly affect behavior and influence every decision and action that we make. It's also important because the world is full of cues that push and pull our feelings and that limit our ability to choose freely in the repertoire of actions that are afforded to us. This is a highly technical diagram, by the way. Those cues can be things like that tasty beer that I might have had last night, or chocolate cookie, or this cute mama spider with her babies, or things that are happening in my body, pain and itch and nausea, or really just about any cue at all. So life turns out to be full of emotion, um, things that we want, things we fear, things we want to avoid. They imagine that you're on your way to deliver your dissertation and you're carrying your papers and you get bumped in the hallway. That becomes a very emotional stimulus. All of these cues here, you see the papers flying. These are affective stimuli now. The pages falling to the ground. You're late. Um, this person's face, is he sorry? Does he care? And, what is this guy doing over here? That's a very affective stimulus. He's walking away. So it's everywhere. Um, and in addition, our feelings feedback and affect learning. So for example, with a cue like this, this mug of beer might be a cue that can elicit cue-induced craving even many years later after I've been abstinent for a while. And that was shown by a recent meta-analysis by Hedy Kober. So we're constantly learning about those cues, and those really essentially enslave us because they constrain our choices. And reappraisal or regulation of emotion is one of the ways in which we can free ourselves from that. And there are many ways of talking about emotion regulation, but I want to talk about it in this way, in the way of changing attributions. And so here I have two different stories that I can tell myself about what happened to this person. I can say, he's so rude, he wasn't looking where he was going. Or, and that's a character attribution, or I can make a situational attribution. As Dick Nisbett, sort of studied many years ago, he's nervous, he's running late. Let's acknowledge the uncertainty in that situation. So that's really changing the answer to why I'm experiencing this. And to do that, I'm operating with counterfactuals, things that I don't know and can't know that are hidden from me. So I'm filling in information, right? And that really is what affords flexibility. So is there an alternative causal attribution for behavior? Um, okay. I'm sort of one step behind on the slides there. I'll get it. All right. I'm talking like there. All right. So there's three parts to the rest of the talk. I'm going to start by presenting a canonical view of emotion regulation. So very old school. I'm going to talk about neuromarkers because I think they're really important, how they can help us to gain leverage on what's affected. And then I'm going to introduce another tool as well, and come back to this classic emotion regulation paradigm, but with a new analytic tool. So to start with, a convenient starting point for many of our thoughts is this kind of stage model of how perception and emotion works. So there are perceptual systems that feed into value and appraisal systems, and those lead to action. And we might be able to associate different regions, like visual pathways with perception, places that we love, like the amygdala and the nucleus accumbens with value and motivation and affect, and other circuits like the anterior cingulate and motor cortex with action. And I think that that's not right exactly, but it is a useful place to start because that lets us try to dissect that system. And that's really what I'm getting at today. If that were true, I might be able to identify bottom-up influences that are targets of regulation that are producing affect. I might be able to identify things that are activated in the brain that provide the regulatory signals and respond to context. And this model really works if regulation and generation are really separate things. And we can measure each one, and then we can study the effects of one on the other. Okay. So the classic paradigm looks like this. I've got this spider, and I'm going to compare that to, this is an aversive image here. I'm going to compare that to neutral images. And if I think of that as a cute mama spider with little eggs, then it doesn't feel so bad, right? I've generated some new context, and I report less negative emotion. And that drop in emotion is reappraisal success, at least as measured by self-report, and all of its inherent limits. When we look at what's happening in the brain, so this is an old study, but I think it's still quite useful. I think it's served a lot of good purposes. There are areas in frontal parietal areas and ventral frontal and temporal areas that are activated, and the more they're activated, the more reappraisal success people report. And we can look at pathways from the cortex to the subcortex. So the more activity during reappraisal in the lateral prefrontal cortex, the more activity in the nucleus accumbens, and the more activity in the nucleus accumbens, the better people report feeling. And if we control for that pathway in a mediation analysis, we can also look at the amygdala. And it turns out the more activity in the lateral frontal cortex, the less activity in the amygdala, which makes us feel worse and we feel better. So this is really a nice, convenient model. We can also search for all the areas in the frontal cortex that have that relationship with the nucleus accumbens. And we see there are multiple areas in the lateral and medial prefrontal cortex. And by the way, we replicated this mediation finding recently in a study of 350 people, which I won't show you that result, but I'll show you the study at the end of the talk. And we can reappraise different kinds of affective stimuli in the same way with potentially similar effects. So if we reappraise pain, the idea is we activate these regulatory circuits, whether we're using our thoughts to turn pain up or down. So that's what you see here. And we also might activate our nucleus accumbens in a valenced way. So it goes up when things are better and down when things are worse in this case, in this study. And these kinds of findings and many others have led to what I call the canonical view of emotion regulation, which this is one view from an older meta-analysis, but there are a whole series of meta-analyses now. So when you're regulating, you're activating the prefrontal cortex and the TPJ and the parietal cortex that you see here. And you're deactivating really one place in the brain consistently, which is the amygdala. But it's really not always consistent across studies, actually. And there are variations by regulatory strategy in meta-analyses. So when you're generating new context by reframing events, that's the red that you see there. So that's most consistently activating this network. There are also suppression, not so good. Aha, okay, good, I'm keeping up. Then there's also hypoactivation of this network in patients with anxiety, for example, in another meta-analysis. So there's clues that the regulatory process is disrupted. Okay, so that's a drop on the classic view. I want to sort of say very briefly that I think that canonical model isn't sufficient, and just a drop about why, although I'm sure in this time I won't do justice to it, and talk about two helpful developments, predictive models and the use of Bayes factors for system identification, which I'll get to at the end. And both of these kinds of techniques in general require larger data sets with more diverse tasks. So we have to put together data across many different tasks and conditions to really to do this properly. And especially with the Bayes factor analysis, we actually need larger samples. So the kinds of data sets that we're now starting to collect in the hundreds or more suddenly become very useful. And then we can do things that we just simply couldn't do before. So this nice view that I said doesn't have to be the case. And in fact, it's not. Many neurons in the brain, if you look at neural populations, they display mixed selectivity. They respond to many different kinds of events, and there are different populations of neurons all mixed together. And if you look at how artificial neural networks are trained to track current motor coding, for example, again, the neurons that are tracked by these artificial intelligence models have mixed selectivity. This is the work of the Churchlands and Mark. And they have very, very complex populations. So it's not as though there's a clean separation of action and value and different kinds of movements and pieces. It's sort of all intermixed in one big recurrent neural network. So that's certainly how things could be as well. And in many ways, I think they are. And if that's the case, then we can't really separate generation and regulation. And we can't really study the effect of one on the other. It becomes very difficult. And I think that's kind of where we've been as a field, as having difficulty separating these things, which is sort of a motivating theme throughout this talk. So how the brain really encodes affective information, as we've learned tons about now with animal neuroscience studies, is through distributed patterns in local circuits. This is one example of some beautiful work by Greg Corder and Greg Scherer in Science a few years ago. And they are doing calcium imaging and recording of populations of neurons in the basolateral amygdala. And what I like about this study for this purpose in particular is that they studied lots of different things. There are different populations of neurons that respond to noxious heat, noxious cold, pinprick, mild touch, light touch, chocolate, and shocks, and other things. Okay, so they have all these different populations, and you can see that there's really a mix and match of these neurons that respond to different combinations of these different things. Okay, so lots of overlap within the amygdala. This would all be within one or a few voxels in the brain. So how could we hope to really disentangle that? But it also turns out that those ensembles are connected to other ensembles in other brain circuits that are remote. So now we have distributed ensembles too. And the hope is that we can use patterns of information both within areas and across areas to get more specificity and sensitivity in what we're measuring. So this is one way among many to outline the specificity problem, if you will. This is from a meta-analysis of a few years ago of 150 studies that were handpicked to evoke emotional feelings of different kinds in relatively category-specific fashion, nominally. So there's anger, stress, fear, happy, sad. And what you can see here is that there's significant activity in the amygdala for all of those categories. And it's not just that, there are these broader patterns as well. So it turns out that we can use these data in a meta-analysis from some studies, the coordinates of some studies report, to decode or predict which of the 5 categories other studies are studying. And we can do it with, to me, actually shockingly high accuracy for this kind of messy, very, very messy data, which is about 60% accurate classification. But we don't do that based on the amygdala. We do it based on the whole pattern across the cortex and also the pattern of co-activation across these different areas in this case, like the connectome, if you will. Okay, so there's a lot of information in that connectome. So the idea is that we want to go from brain maps where we have a task and we're looking at one voxel or one region at a time. So what is the amygdala doing now in response to this or this or this? To a different framework where we've reversed the predictors and the outcomes. And now we have multiple voxels together that jointly predict an outcome like threat or fear or pain. So that really turns the model around. And then what we're doing is we're using the brain to make testable predictions about behavior that can be falsified or validated, hopefully across studies, as I'll show you. And importantly, for this talk and all the work that we're doing, we've trained models where every person gets their own individual pattern. That's a very strong theme in neuroscience, in human neuroscience. But here, we're always training models on some individuals, identifying a population level code, and then transferring that to other individuals and other complete cohorts and data sets without touching the model or retraining it so we can validate it prospectively. And if that works, that's a very powerful way to go, as I'll show you. And one of the underlying principles is that we have these voxels that are really very large and coarse, and we have intermixed populations of neurons. But randomly intermixed doesn't mean uniform. And so even within a region like this, let's say I've got two different types of neurons that respond to two different tasks or functional properties. What we're tracking is something often roughly related to the local field potential. So I have a pattern for neuron type 1 and a pattern for neuron type 2. I sample this very coarsely with my voxels. And what you can see is that there might be no individual voxel that shows a very strong signal at all. They're very, very weak. But as an ensemble, the patterns are reliably different and differentiable. So that's sort of one of the principles. And the second principle is that we're going to borrow strength across the whole brain. We're going to try to identify all the areas that show those signals together and average over them, essentially. And this is a way of approaching a problem that I think we should talk about in the field, which is some of you might know this paper by Sean Merrick and Brenda Turber-Clemens et al. That appeared in Nature last year. And the tagline was that you need thousands and thousands of people to detect any reliable effects. And this is true, I believe them, if you look at individual voxels one at a time, and you're looking at individual differences in complex phenotypes that have lots of issues in terms of reliability and other measures. And so here, I just want you to focus on this, which is the correlation between the brain and behavior. And that's 0.1 at best, basically, right? And these are all different phenotypes. So I think this is a measure of general fluid intelligence. Mental health measures are down here somewhere, right? So they're really small measures. But now look at the y-axis. If you use a whole brain predictive model, you can do a lot better. In fact, you can do the correlation's 4 times as strong, about approximately. And that may not seem like that much, but if you think about powering your study, if you do a little simple power curve with R equals 0.1 for your association, indeed, you need 3,000 people to achieve 80% power. But if you have an R of 0.4, then you need only a few hundred people. And we made this point with many more analyses in a response to their paper, which was, I would say, not well received by them, but I won't show you that. All right. So today, we're going to take, then, the logic is a discovery validation approach. And that is that we want to identify models based on these patterns. It could be patterns of activity or connectivity or graph metrics, as Olu was showing very elegantly yesterday. And the things I'll show you, actually, we have preferred patterns of task-evoked activity because we think that they're relevant for behavioral measures and they're easy to interpret, and you can actually take those regions and pathways and go back to animal models and try to cross-validate them, or you can stimulate them and stuff like that. So they're just transparent. We've done things with other more complex metrics as well. And then we want to validate the heck out of it. We want to characterize how sensitive is it to the thing it's trying to measure, how specific is it, and refine it, look at its robustness, try to interpret it in light of other findings. And when we have a provisional pattern that works well, and this works especially if I can do this across individuals so that I can test more studies and bring in more data, then I can use that to test interventions. So it could be a drug, or even better, a placebo. It could be a psychosocial interaction and the effects of psychotherapy. It could be brain stimulation. I'm gonna put the brain stimulation in there just for you, Alvaro, or other things like mindfulness meditation, for example. So this is one of the first such signatures, and Richie mentioned it, and I think it's useful because it illustrates the process, but it's also the most widely validated signature. So it shows you more about what we can do with it as we gain more data. But I won't spend too long on it because I'm going to go back to emotion regulation soon. The idea is that we take a sample of people and we're going to do a manipulation, in this case of evoked experimental pain, and we're giving them 4 levels. And we go from barely painful to about as hot as they can stand. And we're varying that pain then within person. And then we're using a giant regression model that's stable under many, many variables. So it's just some machine learning tricks to identify a signature, and that's a pattern. And that's a set of weights. And then what I can do is I can apply that set of weights to any brain image and get a predicted pain response. The pattern was called the neurologic pain signature. It involves activity in the dorsal anterior cingulate, anterior mid-insula, medial ventrolateral thalamus, S2 and posterior insula, all areas that you can expect to be the nociceptive afferents, and a lot of converging work, to make a long story short. So that's great. But it's not just that there's activity anywhere in the anterior cingulate, which is not specific. It's there are particular places in the anterior cingulate in these other areas, so that we have a fixed target and it's not a moving target. That's really important because there are many such models that are being trained. But here's a litmus test. You have to be able to take that model and apply it to a new person and make a prediction without knowing anything else about the outcome. And a lot of the models that are coming out, that's not very easy to do. But this one, it is. So we can take a new sample, take people one at a time, take any person, and we can apply the signature by just simply taking the weighted average. So we feed in their data image. It might be a task-evoked response of some kind. And that boils the whole brain image down into one number, which is the predicted pain for that person in that condition. And again, we're always going to test models in new individuals. Okay. And if there's accurate prediction in this case, that really also means that there's a consistent brain representation across individuals that I can use, which is also really important because it doesn't have to be the case. When we started this work, we thought maybe every person has a very different, pain system. Why wouldn't they? I suppose, right? Maybe you develop it differently with, plasticity. But it turns out that this kind of pain system, at least, is remarkably conserved across individual people. So you see here, each line is an individual person, and there's the individual levels of stimulus intensity. So that works. And then we can take that and we can feed in images, and we can do a classification test. We can say if we had to pick between two images, and one of them is painful heat and one of them is non-painful warmth, or one of them is recalling pain, or one of them is anticipating pain, we can ask how accurate we are in classifying that. And here we're about 95% accurate in this forced choice test across individuals. So that tells us we have a strong signal, and it's specific, at least in this case, for pain. And then, because this works across people, we can scale that up. And we've had a really wonderful time testing this on data sets from across the lab, probably across the world, about 50 published cohorts of people at this point, and growing. And the AJP paper was a wonderful example of that using meditation. So that was a lot of fun to do with Joe. And what we found in a nutshell, and there's a lot more to say about all this stuff, but I'm going to give you some of the highlights, is that this responds reliably. It's sensitive at the levels I showed you to lots of kinds of brief evoked pains. Not so much tonic pain that lasts a long time. That's another story. Okay, and more stuff, right? Okay, there's some things that are partially confusable with pain, but they don't seem to activate to the same degree, or no. And it's not activated by a lot of things that have been in the published literature talked about as activating pain circuits and being confusable with pain, including seeing the person who rejected you in love, seeing other people in pain, and other things that we've talked about here. And this is just one example of this, where we are starting to put together larger and larger data sets. So this is a small one now. Actually, it's 270 people, but they're systematically sampled across different studies, where we have 18 studies, and they're evenly divided into 3 domains: pain, cognitive control, and, ooh, that shouldn't say pain on the blue thing there. That should say negative emotion or negative affect. And then 3 subtypes of each one. So we have coverage, right, of each. Now what we can do is we can look at sensitivity and specificity and generalizability. So what does this pain signature, so-called pain signature, respond to? So here, what you see is each bar is a study and each dot's an individual person. And you can see that it responds across multiple types of visceral pain and thermal pain and mechanical pain on different body sites. It responds for virtually every person, at least in the right direction. And it doesn't respond much to these other kinds of challenges, including social pain. And this is, except for one study here of sound, right? So there are some other patterns we developed since then that are sort of more specific against aversive sounds, but by and large, it works pretty well. Okay. And I just want to point out too that the effect sizes here for detecting states are now 10 times larger than those that are in the largest effect size in the BWAS paper. And that's important because it bears on the kinds of strategies that we might want to use to understand relationships with psychopathology between person. So I would characterize a lot of what's being done without casting any aspersions on it at all as the direct approach. You take a patient category or symptom category, you try to predict that thing with the brain, right, with machine learning, AI, and stuff like that. And what happens then is you're fighting all of the problems with reliability, you're fighting problems with diagnosis, you're fighting problems with confounds, with medication and sleep and diet and exposures and all these things that you might be detecting instead of the thing that you're interested in, the condition that you're interested in. So what we're exploring here instead, and I won't give you a lot of clinical results anyway, I'm going to talk about emotion regulation, but is this idea that we can try to find things that are relatively homogeneously encoded in pain, like in the brain, like this kind of pain or this kind of threat response, and then we can develop markers for those processes. Now those ought to relate to symptoms and clinical disorders in a many-to-many kind of fashion. Right. So one of the ways we can use this is to compare different kinds of interventions. So if it's pain, there are many things that I can do to establish an equivalent dose of pain relief. And it could be morphine or oxycodone, but it could also be EMDR, or it could be aromatherapy, or it could be psychotherapy. It could be a lot of different things. We don't really think that they work the same way. We don't think that they all have the same impact or should have the same reimbursement status. So we can start to use this to understand which things reduce activity in this particular pain signature and which don't. So I'll go to one of my favorite subjects, which is the subject of our talk today, which is cognitive reappraisal. So we can see here pain readings go up reliably across stimulus intensities. And when I instruct people to reappraise some of these stimuli as burning, bubbling, horrible, then the pain goes up. And when I ask them, that's the yellow line. When I ask them to reappraise the stimulus as warm blanket on a cool day, spread it across your body and you feel safe, and pain goes down. So that's great. David Spiegel tells me these are like hypnosis instructions that he might give, although we didn't hypnotize people here. So then we can ask, what happens with our pain signature? Is it sensitive to this? And you can see that it responds reliably across different intensities. So what happens with self-regulation? And the answer is nothing at all. You can't self-regulate this. So what that tells me, first cut, is that there's a stage, this signature is reflecting some stage that's pre what you can do with this particular strategy. And whatever you're regulating comes after that, more on the decision phase side, value construction side, or something else. So now I can actually use this lens, and this is why partly I wanted to show you the NPS, because this doesn't exist for any other signature yet. I can look across 34 different studies, and I can look at what happens with 20 different studies of placebo effects. And the answer is nada. There's virtually no effect. Effect size is 0.07. Okay, smaller than the BWAS. But I can look at other kinds of cognitive interventions, like giving reward right before pain, manipulating perceived control over pain, which I love, and cognitive appraisal. And the answer is nope, not an effect. But you can see that the graph is going up, right? So there are some things that actually work. If we start eliciting states of social touch, so for example, a woman who's having her hand held by a romantic partner while being scanned with pain, that induces a safety state that's more sustained. That has a significant reduction in NPS. The positive here means reduction. Other forms of conditioning, so presenting expectations reinforced by cues, can, under certain protocols, produce a significant reduction. And these are two studies, one published and one unpublished, that we're writing up on mindfulness. And they produce larger effects than any of those things, actually. And I was going to try to put slides in, but I couldn't do it. So I'll spare you the data. I'm sorry, I love it. But in the one of those, we actually did try to hypnotize people first, and then induce a state of safety and receptivity. And then we gave them instructions that your pain can modify, brain can modify your pain, and that they can separate the hurt from the pain while we hope they're in that receptive state. So that's promising. And the only other thing that's better is good old opioids, right? Okay. So that's a little bit about comparing interventions. It's not just pain. We've actually had a little cottage industry. We've been going crazy and developing neuromarkers for all different kinds of affective states, especially brief evoked conditions like this. So you can see many of these here. Many of these I hope will be of interest to the group. I'll talk very briefly about a few of them and some of the things that we've learned from that. And all of these things are available, and they've all been validated out of subject, and many of them, but not all of them, in entirely different cohorts. And that's a work in progress as well. We like sharing, so they're all on a website. You can download them and you can try them out and you can use them. We have some tools to help apply them easily. So the idea is that we can apply them to existing data and we can accelerate the cycle of development and validation. Let's find out what works and what doesn't work and scale up the things that work. All right. So maybe we can apply these to Raquel's 9,000 person data and see what happens, right, with some of her tasks, with a threat signature, for example, right? Okay, so I want to say 4 things I think we've learned about the coding of affect in the brain or our learning, and some of them are surprising. First, there's no one brain system that's necessary or sufficient to predict pain and other kinds of affect. Amygdala doesn't work so well in human imaging. Doesn't mean it's not super important, you know, but it's not a great signal. Secondly, as you'll see in these signatures, cortical areas are often more predictive than subcortical limbic areas or amygdala or nucleus accumbens, depending. So there's a lot of direct correlation between cortex and different kinds of experience, trial by trial. Third, when we look at these different kinds of affect, threat and fear, they're really all reliably different in the brain. So they're very context specific in general. Although the NPS is the most generalizable in terms of many kinds of evoked pains on many body sites, it turns out, of all of them. And 4, which I wanted to really include because this is something that Carrie touched on the other day, is that I think that we have more evidence that affective responses, how you're feeling, is predicted by activity in sensory systems, as well as these sort of traditional limbic or multimodal systems as well. So if we go back to our cute mama spider, and we just want to be able to predict from the brain, how negative do you feel right now on a scale of 1 to 5 from this, when you see this picture or others like it, we can train a model that predicts it pretty well. It's a very, very large effect size here. And you can see there's a cross-validation sample, and then we have a totally independent holdout test sample where we get the same thing, right? So we haven't fooled ourselves into thinking that it's better than it is in this case. Okay. And that signature looks like this. It involves activity in the amygdala. It shows a hot spot there in the anterior insula, in the dorsal medial prefrontal cortex, and precuneus, among other areas as well. And the point I wanted to make with this, in part, is that if we look at, we have a, let's see, we'll see if the graphics kind of show up here. But if you just look at the amygdala and ask how well can you decode what they're feeling, you can. It's significant, but it's a very modest effect overall. Same with the anterior cingulate, the same with the insula. You can take the best single region across the entire brain, which is biased because you're capitalizing on chance. It's better, but you can take this unbiased predictive model and it is much, much better. So that gives us a signal that we can study the modulation and interventions and how it performs. Okay, so I want to show you briefly 2 other threat-related predictive maps, which are surprisingly different than that. And that was trained on a mix of very aversive pictures, some sort of gory scenes, essentially of bodies and things like that. This is another signature that Marianne Redden and colleagues with Daniela Schiller trained to track evoked condition threat. So the CS plus is a auditory cue paired with shock. CS minus is a safe cue, as many of us know. That elicits reliable skin conductance responses in this study. We can look at what happens during extinction. Let's see if they're, Okay, so here's the pattern, and you can see the amygdala is involved kind of weakly again. The PAG there is the sort of big hotspot, periocular electric gray, which is also really important for threat. But there's a lot more involved as well. Okay, so then we can look at what happens during extinction, and we can look at what happens to the threat expression, right? And this is if you get an actual extinction protocol where you get the tones alone, or I'm sorry, this is no extinction. That's, so there's still a response, right, to the CS plus during extinction. This is real extinction, where you get the tones alone. And then this is one where you just imagine the tones. The extinction cues are totally imaginal. And this works just about as well, right? So this is, I like that, I love that little result. So it just relates to what, I think Edna Foa and many others did with imaginal exposure before you go to real exposure. So it can reduce this signature. It also reduces skin conductance responses in the same way, and the two are correlated in this case. Okay, that's a drop on that. But now you might think, okay, so that's a great threat signature. That's going to capture, a good space of threat and fear. But a response to a CS plus is not the same as fear. So later, Feng Zhu and Ben Becker took a much larger data set. I think this one has a couple hundred people in it, and if I'm not wrong, and trained and delivered a variety of images that are elicit a variety of levels of subjective fear, like this cute hairy tarantula. Okay, and here, so this is a pattern that is a multivariate pattern that's predictive of how much fear people report. And you can see rating 1 is the low fear, and across 5 levels. You see a really nice evoked response here. There's 3 different tests, a cross-validated discovery cohort, and then 2 other separate data sets. So it seems to really work for this kind of paradigm across data sets. And I don't have a graph to show you, but what's really interesting is that these 2 signatures are doubly dissociable. So they tested in other data sets whether the threat CS plus signature tracks fear, it doesn't. Whether the fear signature tracks threat response, it doesn't. And both of those are at least partially dissociable from the signature I showed you, the PINES, picture-induced negative affect signature. So all these things are trained in slightly different ways to capture something that's related to the same broad construct, but they are delivering reliably distinct results across people. We've been doing stuff too in the positive affect space and reward space. I won't have time to show you a lot of stuff, but this is one on sexual image processing with some collaborators in Germany and the Netherlands. We don't study those at Dartmouth now anymore. Sorry, I shouldn't. I'm gonna self-regulate. And so this is it. Okay, so like there's yellow means greater, sort of greater response for sexual images, and blue is a decreased response for sexual images. I want to point out a lot of overlapping areas like the nucleus accumbens and PAG. I also want to go back for a second and point out this high-level visual cortex is predictive here. Some part of high-level visual cortex is predictive here. And we're starting to see a theme. And same thing with the PINES, the other response. Okay. And I wanted to pick this one out in particular because you can see, again, you might think, well, this is any kind of positive image or something. I might have thought that, but it's really not. And it's not just any kind of arousing image either. So what you see here is this is a cross-validated discovery cohort of 100 people. This is responses to sexual pictures. Each is an individual person here. This is positive images. There's really a big difference, not much response. These are aversive images. These are neutral images. So there's nothing that looks like sexual pictures. This is an independent replication cohort where we find the same result. And if you zoom out and look at all of these different signatures, then they all actually look different. Pretty much. They're mostly all uncorrelated. And there's other ways we can test that, and I'm happy to talk about that. But as a first cut, we think that they're really not measuring the same thing, but they are measuring something that's reliable across people related to a particular context. We can also use these to train and test what's the value of these networks. Maybe all of these things map onto those resting state networks, maybe in a simple way, but they don't. So this analysis, developed by Phil Kriegel, looks at different numbers of voxels trained within individual resting state networks. So only the default mode network or only the ventral attention network, et cetera. And you can see that you get performance everywhere, but nothing works as well for predicting pain as the whole brain model. So you really need multiple networks working together. This down here, by the way, is the best single region in the brain, which is far worse. And so we can do this for lots of different things. We can look at pain. We can look at our subjective fear signature that I just showed you. And you can see the whole brain model dramatically outperforms any of the single network training models. We can do it with the sexual image processing. We get the same kind of result. Okay. I'll pick out one more example from this section that I think is really interesting for a couple reasons. And one of them is that we really attempted to put together many kinds of affect. So we started doing something that I would have advised people to never do, which is have 25 different conditions with 4 levels of intensity of 4 different kinds of affect, plus some other pictures of cute bunnies and stuff thrown in, which you'll see in a minute. And so then what we're going to try to do is use a partial least squares model. I'm skipping the methodological details, but to simultaneously develop signatures, brain patterns that predict across individuals just how bad it is on a scale. You can see aversiveness ratings to all these things, regardless of what the stimulus type is. And also, can we detect things that respond, that correlate with aversiveness in a trial by trial way, but only for one type of stimulus, something that's sensory stimulus type specific? And the answer is, this surprised me that we can do this. I actually thought we wouldn't be able to do it for real. But you can see here, this is all, again, cross-validated. So it's out of participant, so it's fair. And you can see that this signature tracks the subjective ratings of aversiveness. Doesn't matter which type it is, really. And this is what that pattern looks like. So you can see many old friends, like parts of the PhD and intersinglet and so on. But again, we're sort of interpreting and we're using the whole pattern together. There are many ways of testing this in an open-ended way, but one of them is to apply this to cute bunny images, so pleasant images. And you can see that pleasantness levels go up across different levels of pleasant images, but the pattern response doesn't. So there's some specificity to valence here, at least as a first pass. What's interesting too is that we also find for every one of these other types of responses, I should say what they are. There are 2 kinds of pain. They're mechanical pain, the stimulator on your thumb, thermal pain, heat on your arm, aversive sounds. It's a knife on a plate. It was the most aversive stimulus from the IADS, and we give it different levels of loudness, and aversive images like getting your wisdom teeth taken out and various things like that. Okay, so now we have these models. We have 4 different models. Each one tracks aversiveness, and here you can see it responds, but only for that, that stimulus type, and it doesn't respond to the other stimulus types. We replicated all of these in independent data sets. I think we have 8 different cohorts in this, this study, but I'm not showing you that here for time reasons. And those models look like this. There are specific, at least I'm just focusing on specific hotspots that are the most reliable. And what you see is for mechanical pain, for example, lateral somatosensory cortex is particularly important. And that tracks a lot of what we're seeing with chronic, with evoked back pain and chronic back pain. There are different areas that are really hot spots that are selective for thermal pain in parts of the anterior insula and posterior insula and medial dorsal thalamus. The auditory model and visual models have parts of the model activity that's predictive, not only in early sensory cortex that's modality selective, like A1 for auditory and B1 for visual, but also some of the subcortical nuclei that send projections to those, like the inferior colliculus for auditory and the superior colliculus for vision. So that's another clue that there's some affect that's correlated, at least with what's happening in these sensory pathways. And we can look at any these sort of different macro level regions, like the amygdala, for example, and we can decompose that it's really a mix of common general aversiveness to lots of things and a contribution from a visual specific model. So that's the purple and the blue, and you can do this for these different kinds of regions, but every one is sort of a mix. It's not only general, it's not only specific, but there are combinations, right? It's contributing something to multiple things. And you can turn it around the other way and say, okay, well, let's say we want to predict somebody's self-report with a combination. Do we use this common pattern response, or do we use a modality-specific pattern response, or do we use both together? And the answer is, for all of these, both together. They both contribute. So the idea is that what you report as aversive is a combination of some general system for aversiveness or threat and something that's specific to that particular stimulus type in that context. Okay, so where that leaves us is this idea that if we want to use neuromarkers to evaluate various kinds of interventions, I have to choose one or develop one that's appropriate for the context and type of stimulus and population, right? And that might be one that exists and it might be something new. And so here's a library of those. And then you can use that to test your interventions, right? And often what we'll do is we'll build in validations within the study so that we know that it's happening, right? That it's working in our study as intended. Okay, so now let me go to the last part and come back to the reappraisal of aversive images. And now I'm going to use this data set from Pete Gennaros' lab from his program Project Grant, which he wonderfully shared with us, and it's been a wonderful collaboration. There's two studies that recruited broadly from Western Pennsylvania with almost 200 people with usable data in each one. And the design is the classic old school emotion regulation task. So what happens is people get a cue that says, just look at this stimulus, or reappraise what's coming next, make it less bad for you. And then they see either a neutral stimulus or an aversive stimulus. So if they're just looking, they see neutral and negative stimuli. If they're reappraising, they see a negative picture. And then they make that rating on a 5-point scale. So that's a little bit about how the task works. And behaviorally speaking, it works. You can't see the individual relationships here, but virtually every person regulates and they report a drop, at least in negative affect. So that's the emotion generation related signal and emotion regulation related signal that we're going to look at. And we could decompose the strategies they use. They use a variety of different strategies that Kateri-McRae cataloged some years ago, from things like injecting new context to change the circumstances, saying it's not real, changing your appraisals of the agency of the people there, and other kinds of strategies. So we can't really sort of fully control what they're doing, but none of them actually really fully mattered in this case either. Okay, so now we can look at this pattern response. I'll use the pines because that's the closest thing. It's a very similar, in fact, it's the same almost stimulus set in the same types of images. So then we can look at what happens with the pines when you're just looking at neutral pictures and when you're looking at negative pictures. And again, a very strong response. Most everybody shows it. And then what happens when you're reappraising? It goes down. Yay. A little bit, right? So that's the negative emotion effect. Again, virtually every person, but a couple in the sample is going in the right direction. There's a modest effect here. Effect size of 0.4. It's highly significant in the sample, but it's a pretty modest effect. Okay, so you might conclude from that that things are looking good for reappraisal of negative emotion. I can also apply this to the general and visual specific negative affect patterns that I just showed you. And so what's interesting is both of them actually, negative pictures activate both patterns versus neutral ones. So that's great, very strongly. What happens with negative affect is there's no decrease. It actually goes up. So whatever people are doing is not decreasing that pattern, but what it is doing is decreasing this visual specific negative affect pattern. So we think then it's having this effect on the sensory specific affective pathway, but not the modality general one. And that's going to fit in with what I'm going to show you next. Okay, so that's the idea. We have some selective effects of this. Now, I want to make an admission to you. I think this is a good step, but this doesn't really answer the big questions yet. Of course, big questions are hard to answer. Including the question of where exactly is the feeling, and how separate are reappraisal and the systems for generating emotion. Okay, so we don't know how separate they are. And for that, we need a different approach that's, I think, complementary. And that's the Bayes factor approach. And we're starting to do this more and more, but we're starting with this idea that often the things that we'd like to infer about a brain region is not one particular contrast or comparison between conditions, it's rather a conjunction across multiple different conditions. And here, and this is a very simple data set, so we have a very simple-minded analysis. But the axiom is that any negative emotion regulated system that's downregulated should A, respond to the negative images, and B, decrease when you're reappraising. And we can argue about that in the discussion section if you want, but that's a first pass. And so then we can take all of the regions that respond and we can divide them up into two kinds of regions, a kind that is not affected and a kind that is affected, right? Non-modifiable and modifiable. But importantly, this is a conjunction of a positive finding and a null finding, which is sort of awkward historically with p-values and so forth. But I'm kind of hot on the idea right now of doing a Bayesian t-test. And what that is a statistic that can turn, with some assumptions that we often make anyway, a t-value from a contrast into a Bayes factor, which quantifies the evidence in favor of an effect or a null effect of a certain size. So here, what we're going to look at is a Bayes factor with a cutoff of 10-to-1 evidence or strong evidence, either in favor of a real change, a decrease, if you will, in emotion and response, or it's more likely to be a null effect than a moderately sized effect with the 10-to-1 odds ratio. So that gives me equal, sort of putting these hypotheses on equal footing. And what's interesting about this is that you can only do it with large sample sizes because your t-value can never be less than 0. And if your t-value is 0, there's this lower bound in the Bayes factor right there. And that's determined by the sample size. So with 150 participants, then a t-value of 3.15 or greater supports the alternative hypothesis and effect. T-score of within 0.43 is evidence for a null effect. And if I have a smaller sample, then I just can't get enough evidence for null effects, essentially. Okay, so we can use that to look at what's regulated and not regulated. And we can also use this to test and understand the overlap between emotion generation and emotion regulation. And so it's fairly intuitive and plausible that they might really overlap a lot. Because as I've argued before, any stimulus can be affective, and it's really your appraisal of that stimulus that makes it what it is. And so maybe the same cortical and subcortical machinery that you use to look at a picture and go, oh, that's really so bad, is the same machinery that you use to say, maybe it's not so bad. Okay. And that makes sense. And this is, James Gross and Lisa Feldman have written a really nice sort of piece on that. So there's our emotion generation and regulation. And what's common to both, any system that goes up when you're looking at something negative and goes up even more when you're reappraising, we're going to call a common appraisal, sort of maybe involved in both systems. And so we can see from historical maps that, yeah, there might be a lot of overlap, right, between those two things. And if that were true, again, you couldn't really study the effects of one on the other. So we're going to use this strategy of the Bayesian t-test to try to test 4 different kinds of brain regions, or try to identify them. And we might find none of them in the brain, we might find all of them in the brain. And one is something that responds only to reappraisal. It doesn't change at all when you're looking at a negative picture. So we know that it's not about the emotion generation. It just responds when you're regulating. There's our common appraisal regions. And there's 2 kinds of regions, non-modifiable and modifiable emotion regions. Okay. If that's confusing, then please stop and ask. Right. I don't know. Okay, and what we then do is take these T maps, which is a pretty large study. They all look really great for emotion regulation and generation. Those are the 2 contrasts. Turn them into Bayes factors and take the conjunctions across them. So this is a map of what looks like reappraise only, and this is a map of areas that are activated and activated even more, common appraisal. And by the way, we also use a threshold where we chose the Bayes factor so that it also satisfies traditional hypothesis testing criteria, and in both studies independently. So we have essentially an intrinsic replication across the 2 studies. And so this is what the results look like. We see the largest set of areas in the prefrontal cortex is common appraisal. So those are the blue. But anterior to those, and in somewhat different areas, there are also areas that are selectively activated for reappraisal. So you can see those in the anterior prefrontal cortex, in the ventral temporal and anterior temporal cortex. You can see them in parts of the TPJ over there. And this is what the data looks like for common appraisal regions across both studies. So you can see it goes up and up again. And then here's what it looks like for the reappraise only studies. Okay. We think then the blue areas, if you were to stimulate them, with TMS or something like that, it might not work very well because you're interfering with mechanisms that are common to both generation and regulation of emotion. What you might want to do is make sure you target the reappraise only regions, right, which might more selectively boost or interfere with regulation. So that's kind of the logic, and that's how we hope that these things are going to be used. They also correlate with reappraisal success. Like I said, the brain-wide association is really hard. Top correlation you'll ever get is 0.1 in a brain-wide search. But here we actually are getting, in a fair test, we just average over all of the reappraise only regions and common reappraisal regions, correlations of about 0.2. Probably can't use it clinically, but there's something there. Okay, and what's also interesting is that we can compare this map to other kinds of maps and increasingly annotate it with what's happening in the cortex. And one of the interesting ones is Daniel Margulies's map of principal gradients of organization from unimodal sort of unisensory input in blue to multimodal input in red. And if you're interested in the networks, the most unimodal networks are the somatosensory networks and visual networks in the brain, and the most multimodal at the top of the hierarchy are the default mode network, limbic, and one of the two control networks. So if you sort of overlap them, you can see it here. They're really right in the areas where they're the most multimodal, the reappraise only areas. And we can also look at those. These are also the areas, generally speaking, that have the largest expansion in some analyses from macaque to human, the largest expansion during development, and and they're at the top level of a hierarchy estimated from macaque anatomy. Okay, so that's the idea. We can also annotate them in other ways by looking at associations with neurochemical binding maps. So this was a really nice set of about 40 PET studies, receptor binding of all different kinds of studies by Bratislav Misic's group. And so they released that. We sort of cleaned it up a little bit, and we can then compare our different components, the reappraise only and the common appraisal, in terms of which kind of binding maps are they most similar to. That doesn't tell us about the neurochemistry directly, but it does tell us something about maybe you want to go look at this system more, or maybe you want to do other studies of this system as well. So when we look at the reappraise only system, surprisingly, because cannabinoid receptors are everywhere in the brain, it turns out that this reappraise only pattern is very similar to the areas that are the highest in cannabinoid receptor density, apparently, which is really interesting to me. And other big hits are mu-opioid receptors down here, and 5-HT2A receptors, which is why I put that on the slide before, and 5-HT1B. Okay, so those are some of the associations, and you can kind of see the overlap there. So that's another layer. Okay, now we can go back and look at which target regions are downregulated. And this is another point of surprise for us. We found that most of these subcortical areas, in fact, all of the subcortical areas were not modified. And there's no correlation with reappraisal either. So if you look at the overall activation, you can see there's really no decrease when people are reappraising. They are activated very robustly by aversive pictures. And we can zoom in on the amygdala and its subregions from a canonical atlas. And you can see the data here in the subregions. And you can again see, we really didn't see any evidence for an amygdala decrease, at least in this paradigm, in this population. And that, of course, opens the door to what would have happened if we'd hypnotized them instead, or done something else, right? And we could test, or are they trained and practiced on it? But that was surprising to us. All of the regions were non-modifiable. The modifiable targets, what are people decreasing, are these higher level visual areas and somatosensory areas. And so what you can do, you can see these side by side here. In a nutshell, I think what's these areas that people are modifying are very close to patterns that are associated with high-level spatial attention and semantics, but not the subcortical stuff. And this kind of helps explain what's happening with the pines, this picture-induced signature. So you can see there's an overall decrease in the signature, but it's not because all the areas are being affected equally. The amygdala is not, the hypothalamus is not, et cetera. They really are driven by these decreases here in these areas. Okay. We don't think it's eye movements. We have a couple of different kinds of control analyses, but they're probably not super great. I know that Richie and Heather Uri have published on this, maybe among others. Okay, so what do we make of this, right? Are these sort of perceptual areas? Well, I think there's a variety of evidence that suggests that they may be more deeply, affective responses may be more deeply embedded, including some of the correlations that I showed you before, some work from Mark Baer's lab that seems to show that reward-related responses can be learned or in early visual cortex. You can get anticipatory responses to reward. And these areas are not just involved in low-level visual processing too. As several analyses, including Alex Huth and Jack Gallant's, these areas seem to become infused with semantic meaning. So they also start to respond to the concepts and the meanings of the words and objects as well. So when people are reappraising, they could be altering the sense of the meaning and the semantics, as well as how they attend to the picture. Okay. And I'll just show you one more thing that kind of goes along with that, which surprised us. And this is an analysis that Phil Kriegel did a few years ago, where he took a bunch of pictures and videos that elicit affect. In several different data sets with thousands of pictures. And he trained a neural network. He took actually a commonly used network, AlexNet, for object recognition, and he added an emotion detection layer to it and trained it to discriminate 20 different emotion category labels generated by humans. So it turns out that EmoNet, as it's called, can do a 20-way discrimination of categories, of object categories, based purely on high-level visual features, because it's not thinking anything or feeling anything at all. So you can see all the category here. This is the confusion matrix. And there were 11 different distinguishable categories that were distinguishable each from the other. And then what he did is he took a sample of human imaging. Oh, let me show you this too. So you can take those categories and you can recombine them. You can train a model with partial least squares to track the normative valence ratings that humans give with pretty high accuracy. So one thing that tells us is that a lot of what people rate on average when they're responding to these pictures is something that is embedded in the structure, the high-level structure of the image itself, right? And we can talk about what that means, but we think that that might actually mean that there are affordances or there's entanglement in these early visual representations that are for affective responses, right? For fast responses in evolutionarily adaptive situations. That's just a speculation. And then what Phil did is took a sample of humans looking at the pictures, and he looked at their occipital cortex, and he had EmoNet give the category ratings of the same, the scores for those. And those two are also correlated together. So you can train a model with cross-validation where all of these models seem to be decodable. All these, the EmoNet's representations seem to be decodable from patterns of activity in occipital cortex. Okay, so that's something that tells us that that where we look for affect might be more complicated than we had previously thought. Okay, so I've talked about three things. I've talked about a canonical view of emotion regulation, which has been useful. I think there have been useful targets that come from it and useful convergence. I've talked about using neuromarkers and patterns to identify brain systems that are predictive of specific types of affective responses that we can then use as target measures in a variety of translational settings. And I've talked about this newer approach of using Bayes factors to really try to identify which features or which brain areas and voxels correspond to certain kinds of properties of a system, including which ones are susceptible to emotion regulation. And I want to end with a few broad parting thoughts because I like wild speculation. And this is this idea, that, okay, so Alhassar and Hochstein have a theory of consciousness and vision that I really like. And what they say is that V1 is of course essential for vision. If you eliminate it, you have blind sight, no conscious vision. but that's not where the vision is happening. That's not what you see. What you see is all the way up in the front of the brain. And it's this construction then. And you can demonstrate that in various ways by change blindness paradigms, for example. So what you're seeing is this high level gist, and you think you're seeing details. And what happens is when you see a detail, you have to attend to it, and you're connecting that system to these early visual representations at some different level of detail. And maybe feelings work in a similar way, so that the construction of feelings is somewhere up here in these frontal and high-level multimodal systems. And we see all these other activations here that correlate with feelings because those are the contents of vision. Those are what we're feeling about, and that is modality type specific. And both of those things are then important for human feelings. And if we think about this as a construction, then maybe feelings really are functions of a more complex state of inference about the world. So, and they start with our projections, our beliefs about what are the potential future harms and opportunities. What are my beliefs about myself and the world? What do I remember? Those feed into a current state. They're combined with our sensory input, our visual input, our nociceptive input from the body or other types of input from the body into this construction of a current state. So it is that that might be how it plays out, and where the pain is or the threat is or the fear is in that state, right? And if that's true, then it doesn't live in any one of these networks per se, and we can measure it maybe as a function of all of them. We can measure reflexes that come from specific parts of the system, like nociceptive reflexes are great. We can index the state with behavior, protection, escape, attack, and so forth. We can index the sort of future-oriented parts with by looking at avoidance, but it really is between all of these different networks. And so this sort of illustrates some of the networks that are involved in connecting this multimodal system in various ways. And my final parting thought, I promise, is that to go back to the idea of attribution. So what emotion regulation is for is to confer flexibility so that we can take the same input and we can construe it in a different way that allows us to choose a different response. Otherwise, we're slaves. And there's something very interesting that happens. So if here's an event, a sensory event, and there's one causal explanation in a very simple like reward learning paradigm or something, you get reward and you attribute that to the cue, right? Then it's all really clear. But if you have two different potential causal models going on, two potential causes, then the optimal Bayesian model does something really weird, actually. It makes an attribution, it updates the belief, what the sensory evidence means, according to how well each of those two models explains the data, according to its likelihood. And that's a combination of your prior belief in something and the likelihood, how well the causal explanation seems to fit that data. And that's called abductive inference. Okay, so this is my future, like, next few years here. What that means is if you have strong prior beliefs that, this person's a, what, right? Then you might get stuck in that belief because any evidence that they do, right? Anything that they do will be interpreted in the light of their, traitorous behavior or something like that. So we studied this actually in this paper here, which I won't talk about, in political beliefs. And so I think this helps explain why it can be actually very rational to have extremely polarized beliefs and even rational to be insensitive to things like election outcomes, because it's always interpreted in terms of your prior beliefs. So that's something that's quite important for a lot of us in the world as we go forward. And I think that attribution and these systems are really a key. So in sum, I want to say I think these competing attributions are a balance point, and that's what emotion regulation does. It supports flexibility. And it may be that many kinds of inner work here support the ability to maintain that flexibility and to reconceptualize the situation, depending on all these different inputs. And I'll leave you with this. Notions of heat and cold, of pain and pleasure are born only of the contact of the senses with their objects. They have a beginning and an end. They are impermanent in their nature. Bear them patiently. Thank you.
Yeah, you stay. Thank you very much, Tor. That was a masterpiece. And now we'll invite up Michelle Margie and Hadley Radlick who will lead off the questions. And Ryan, sorry.
Hello. Okay. Thank you so much for that talk. It was really inspiring to see how you've used like new and developed machine learning methods to create such powerful and robust models that can actually kind of tell us what's going on with our emotions in the brain. And it's just really inspiring. So I have a question about how you can apply your work on neural signatures of emotion to compassion research. And if you can disentangle the effect of, or disentangle what the emotional profiles of compassion might be. And if you've, looked at an individual observing someone who's feeling pain or what their kind of neural signatures look like or their brain patterns. And one more point is if you think compassion towards in versus out group members would be reflected differently.
Yeah, thanks. Well, thank you. There.
Oh, it's good.
Yeah,
Yeah.
fine. Is it better? No, it's not better. Wait. It looks like it's on.
Green light is on.
I'll just talk loud. Okay, I'll speak up. Oh, hey, thanks. Okay, cool. First of all, thank you for letting me talk for so long. It was really fun. Thanks for listening. So I think, yeah, the study of compassion, I mean, many of you are much more expert than I am, but that's near and dear to my heart because I think that that's one of the things that is, it's both so important and it's not going to fall into any clinical disorder. Well, not many of them anyway. It's sort of really between, like, everyday compassion in society is between the cracks. We have a little bit of work on compassion that I didn't get to put in these slides, but ideas. I think that what we were hoping to do, and what I think there's a lot more to do, is to try to use this kind of approach to develop signatures that relate to different kinds of ingredients that promote compassionate behavior or charitable behavior. So in some studies that we did, we identified a signature that tracks people's feelings of warmth and empathy when they see a person in need. So we had these like charity targets, and they were all sort of real stories of people who were in need. And then they listened to these stories and then decided to give up to $100 of their own money, of their earnings, to a charity like that. And so we picked one and we gave it away for real. We gave a lot of money away. And so we have this, one of the, there are multiple independent predictors of this. One of them is this feeling of warmth and tenderness. Another one is a feeling of personal distress, which is sort of has ambivalent, ambiguous relationships sometimes. But in our study, it was associated with more charitable giving. And then other kinds of attributions, like, do you think that they really need the money? Will it help? Do you think it was their fault, that they got AIDS as a baby or something like that? And so all those things mattered, not similarity actually. Of the people gave money to people who were dissimilar to them. They gave money to basically like kids in Africa, and our participants were not. So that, I think that worked to some degree. So we published some work, and it does seem like that, to some degree we can track that warmth and tenderness and affective distress differentially and see how they contribute. I would love for that work, those to be used and like to be tested and further validated. We did a compassion meditation intervention with an app, smaller scale app, but people practiced for a while, and that had a big impact on all those attributions and feelings that I said, as well as their donations. But it didn't have a huge impact on those signatures either. What it really affected was the ventromedial PFC signal. But anyway, so there's a lot more room to kind of develop that, but I think that hopefully illustrates the kinds of strategies that we might use.
Well, thank you so much for the wonderful talk. I would love to speak to you more about your legacy of work on pain signatures and your work on the placebo effect. So yeah, this work has clarified that neural secrets differently associated with nociception from those associated with affective motivational processes. How can these distinctions be used to understand complexities of chronic pain and its comorbidity with depression?
All right. Yeah, thanks. Yeah, we started studying pain. I think in naivete is a good place to start. We thought, there might just be something that tracks evoked pain, and that might apply to lots of different kinds of pain. And it still might. Actually, that pain signature tracks mechanical pain, hyperalgesia with arthritis in the knee. It tracks fibromyalgia pain, and so it mediates if you have fibromyalgia, which is a complex condition, the more if you have that, then your pain signature is higher to a stimulus. That predicts hypersensitivity. The difficulties, there's two interesting difficulties. One of them is perhaps expected, which is if you think about the kind of person who has chronic pain, that's a piece of it, right? It's hypersensitivity to particular stimuli is an important piece. It's found to be an indicator of central sensitization, but it's not the disorder of the pain. So it turns out when you try to look and see if that hypersensitivity response correlates with the spontaneous ongoing clinical pain they feel, it doesn't. And it does not correlate with their
Really
depression.
interesting.
Yeah. What we find is that I don't know how far I go in this, but I'll go a little bit far, because I think what's interesting is that people in the pain field have thought of pain as a disorder of nociception, of painful stuff. There's labeled lines, there's pathways, it's great. But in fact, the person in chronic pain, it's a disorder of much more than that. So it's actually a multisensory disorder. So what we found with fibromyalgia is that they had a different characteristic brain response to just checkerboards and sounds, which wasn't initially expected. And that was reliable. That was 90% accurate at diagnosing case control fibromyalgia. And so they respond differently. They don't like those things, and they're multi-sensitive. And that's coming out in different ways. So now the bigger studies are starting to look at chronic pain as being about more than just a localized pathology or sensitivity in the brain. And I'll say one more drop, which is a different direction. So that's one of the problems, is that chronic pain isn't just hypersensitivity to acute pain. So you need different measures in the brain. And there's nothing really good yet, I think, that captures tonic ongoing clinical pain, frankly. But what we found, this is kind of an interesting example of how sometimes there are unexpected findings, right? So I would expect that with all these different kinds of visceral stimuli and other things that we've tested this on, that if you give people capsaicin for a few minutes, right, it burns, it's clearly painful, then that would elevate this signature. It doesn't. So there's some kind of break point. When you get to tonic sustained levels of pain, the system is different, and there are different things that apply. So we do have another connectivity-based signature that seems to track tonic pain, and that might actually translate to chronic back pain. But that was recently tested in the UK Biobank in thousands of people with not very extensive imaging measures, and it's significant, but it's not very strong. So it's not going to be a clinical biomarker, I don't think. So that's where we're at. We'll see, right? But it sort of illustrates some of the complexities.
That's really fascinating. Thank you.
This is really cool work. I'm really happy, like Richie said, to see this kind of like whole brain, really precision focused use of functional imaging. One thing that I wanted to ask about is kind of something that this has in common with an issue that this has in common with pretty much all the emotion regulation work, which is the use of these like static images and kind of flash emotions that happen really quickly and then offset and are in this kind of experimental situation. They're not quite the same as emotions that we experience in the real world. So do you think that this kind of procedure can address that or work with that in a more kind of time sensitive way?
Yeah, I also hate that. I know it's like, but Pete started this data collection like many years ago, and so that was the design.
Right.
But it's such a big sample, you can do some cool stuff with it. No, I think there's like many of us, are starting to think about more, sometimes you call them ecological designs, so what happens if you're regulating emotion in other contexts during when you're watching a movie, or maybe when you're listening to those stories of real people in need, which is a different kind of stimulus. I think we need a lot more of that. And there are kind of two stories that you can go down. And I think this is like kind of cutting edge as far as stuff that I know about. One is that when you take a lot of these movie data that people are trying to do, so like my colleague Luke Chang and others have been trying to decode valence from movie data when they're clearly like emotional situations, positive, negative, can't do it, fails. They can't find anything, right? So I don't know, maybe it's like going to be really hard or maybe valence, those circuits are too closely interdigitated, right? And there's too much variability across the types of things that are driving valence in the movies for there to be one thing. Yeah. Like they'll watch like a full length feature film, right? Or we have like multiple minute clips back to back of eliciting different emotions, like including compassion or yeah. Yeah, or it would be like an hour or more, 2 hours of continuous movies or stories. The Moth Radio Hour stories are another popular one. So that's one story. There's another. So Wani Wu's lab is doing great stuff in Korea, and they've picked up this tonic pain. So they're doing tonic, pleasurable, and painful things in lots of repeated data sets and publishing some great stuff. They have a paper that I think will come out in PNAS soon that is a valence decoding paper. And so the idea is that they think that they can actually do that in a cross-subject way and get valence specific signals, including parts of the insula and amygdala and stuff like that. So that's, and maybe those stimuli are more homogeneous. Whereas if you're watching a movie and you're sad or happy about lots of different things, then it doesn't work. So.
Tor, so I have many things I want to ask, but I want to just focus on one because we are limited in time. The work with voluntary emotion regulation with reappraisal, we've done that work and many, it's, there are reasons why it's a favored approach because it is tractable. It really can be brought into the laboratory and harnessed for empirical exploration in the ways that you've beautifully shown. One of the things I worry a lot about, and particularly after hearing some of your findings, is, and I know you know this, but I'm just interested in how you might approach it, and that is, this is a process which we would expect to be highly variable across people. And we might expect some people to really be good at it, other people to not be good at it at all. And to plunk people down in a laboratory and do this without any training, would be like, I think asking a person to engage in a complex motor skill without any training. This is a complex cognitive skill that is being requested on demand with absolutely no training. And so, and I was surprised by your finding of a lack of signal in the amygdala. There's so much other evidence where you can see that kind of signal with reappraisal. And so I was just surprised by that. And again, I think in part it may be associated with this expertise. And let me just throw in one other question that is related to this, and maybe you'll have time to say something about it. And again, we're just as guilty of this as everyone. But a lot of, I think most emotion regulation in the wild is automatic. We're not aware of it. It's happening all the time. And we have just failed, I think, as a field to grapple with that. So I wonder what your thoughts are about that.
Yeah, thanks. Those are great insights and questions. I I agree with you, and I was also surprised. And I think that for me, and we looked at lots of different analyses. Like we tried to find stuff. We find correlations with, reports, but not the amygdala downregulation. Um, and there's multiple different interpretations. It could be about the population. So these are people from Western Pennsylvania that are probably much more naive than some university samples who clearly are all like meditators or something. Who knows? So it could also be about the idea that you need some training and practice. So Brian and Denny with Kevin Oxner has done some reappraisal training where if you see the same images again, you show decreased amygdala even to those images and new images. That's interesting. So there's lots of possibilities, right, for practice. And the way I see that, the null finding, and why that's sort of exciting to me is because then that establishes a baseline, right? So what happens then if you could train that population and all of a sudden they can do it and they're different, right? Then so that's the sort of backboard, right, against which you can say, okay, what works better? Yeah. There was, yeah, the automatic. Thank you. Yeah, it's we've been thinking about sort of spontaneous reappraisal, so and things like that. I think there's two versions. One is it's probably and Kevin and other Michael Gilead has done important work on this. Is in psychopathology, it's not that people can't do this, apparently, right? People with PTSD can do it. People with depression can do this on demand, but that they don't spontaneously, right? They don't choose to. Or, a lot of times, I'll tell you what, if I'm mad, I'll do something to make myself more mad. I'll put on the angry music, right? So it's not that, it's the regulatory goal, right? That's different. It's not that I can't use this apparatus. So with a kind of signature like this, I know there's stuff that's coming out. We actually have a detector, and other groups do too, where you could say, okay, are you activating this system? Are you activating your reappraisal only regions spontaneously, in different settings? After training, do you activate them spontaneously? So that's one lens. And I think the other lens is that there's probably really different kinds of processes that I associate with the ventromedial PFC and this subcortical limbic circuit, where we're all the time making sense of every stimulus around us. And that's like implicit emotion regulation. And that's sort of the bedrock, of how we appraise events. And there, I think we maybe need some new task paradigms to look at sort of ongoing spontaneous attributions of events, for example. Thanks.
Hello, Dr. Tor. Princess Felix, University of Michigan. Thank you for your talk.
Thanks.
So I think it's really amazing how your model could encompass so many different types of people's reactions to pain. And also negative affect, although negative affect one was more I don't know, variable. And I couldn't help but think about individual differences in personality when it comes to reappraisal and how one might, like how it was being talked about, instinctively reappraise a negative experience or emotion. For instance, some people's personality, we can talk about people who are more anxiolytic and people who are more impulsive just based on just their personality. And I'm thinking somebody who is more anxiolytic might reappraise something internally, like, oh, that was my fault, I'm the one who did this, and they might like really dig themselves down. Somebody who's more impulsive might reappraise something externally, like this person is this, and they should have done this, and kind of like make themselves more worked up. And I'm thinking to myself, how would this model take into account individual differences in personality that might affect how one views a negative effect and how one reappraises it?
Yeah, thank you. That's a great question. I mean, so sort of two kinds of answers. One is we could look at individual difference correlates and clinical samples, and these are sort of lenses. Okay, if you're high in neuroticism or if you're high in some kind of psychopathology, are you hyperreactive or responsive? So that's certainly possible. So a few tests like that have been done. We don't find basic demographic differences, by the way. Sex and age we always look at. We don't see. So that's one possibility. The other thing that I think is really interesting to look at is even outside of the scanner, but also with scanning, what's the learning dynamics as you engage in a social or interpersonal relationship and you get feedback. So we have a little bit of work that does that. It takes people who are socially anxious and it looks at what happens when they get negative and positive feedback. How do they update their sense of self? How do they then update their impression of the other person, right? So to start to study those feedback cycles. I think that's a really like sort of combining the dynamics of reinforcement learning models with these kinds of emotional tasks. I think is really an interesting way to go.
Hey, that was really fascinating and fun, and actually very, very thought-provoking too. So I wanna, since we have you here, pick your brain and ask you about simulating or imagining the experience of pain, and what you think that might mean relative to your data. So it seems like, it seems that our brains we're really wired, we're really good, right, at like sort of multi-sensory simulations when we're remembering or imagining all kinds of experiences, right? And so really good at mental time travel, and even we're sort of like even wired to some degree to mind wander, but it's much less clear to me how this how we imagine pain. I've been trying to do it myself. I can't do it very well. So so first, how good do you think we are at imagining pain and simulating pain? What does that mean even? What does that mean? What might that mean? And the second thing is getting back to kind of like the heart of your talk. Do you think that we simulate feelings and pain in a similar way?
Yeah, thanks. One of the things that I really hope to look at more over the next years is the power of imagination in different ways. I think Danny Kahneman wrote some stuff about this, but I think it's speculative as far as I know. It's like we're terrible at imagining pain. We can't recreate the experience, and that might create an empathy gap with other people. So I like that idea. We tested a small data set with NPS with imagined pain from Ulf Baumgartner and Walter Leftrida from Germany, and there was no effect. It was great. And my postdoc, Michael Sun, is just writing up a paper where he scanned 10 of us 20 times each. And you can guess where that was over the last years, during the pandemic years. So I was one of the participants. And we tried really hard to imagine pain and in different, real and imagined and stuff like that. And it turns out we can do it. Actually, so we activate NPS, which is pretty rare among all the things that influences that. And maybe that's because, we had 20 days of practice doing it or something. But that was sort of an interesting thing to me. It was kind of eye-opening. So beyond that, I don't know, but it's a good question.
So this was great, and thank you. And you probably can guess where I was going to go with the question. But if you have a signature that you can scale, that you can modify, that you can imply, but maybe not completely prove, that if you actually were to externally modify it, it would translate into a different appraisal or pain perception and so forth. Why not use it as a roadmap for multifocal brain stimulation to actually change the signature and see what happens?
Yes, that's what we need to do. Why have we waited so long? Because it's like, absolutely. I wanted to do this right before I moved from Columbia in like 2000 and 8, we got a grant on it, and then like things happened and we didn't do it. But this is we need brain stimulation because we need direct interventions. We need behavioral and we need brain, and we need to then see, how whether the design of these things are really good for being targets. They're intervention targets. So that's what I want to do more.
And so if I can pull that thread, what about nature experiments along those lines? If you have people with lesions in locations that are encompassed by your signature, do those people then become unable to perceive pain or unable to reappraise or unable keep adding?
Oh, I love that idea. I had never thought of it, honestly. But I know there's some big data sets now of lesion location disconnection things that are available that people have published, like Michelle Tubal-Tashotten. So that would be such a cool thing to do, actually.
Let's talk some more. Thank you.
Great. Thanks. Okay, thank you. Thank you. Thanks, everybody. It's really interesting. Great. Oh, thanks. Oh, go ahead.
Well, it's come to that time, and it's been another fabulous meeting, and what a great way to finish, Tor. Thank you for that talk. It was very, very inspiring and thought-provoking. I want to acknowledge Amber. Amber, you want to come up here for a minute? And we've been blessed to have Amber with us and to do lots of things in our department, but as importantly, this one. And so I just wanted to give Amber an opportunity to say a few words if she'd like. And she's part of the team here, so.
Just thank you all for coming. And please save the date. That's what I'll say. Save the date for next year because it's going to be a really special event. 30 years
Wow.
these two have been putting this on together. So yeah.
Amber, again, thank you so much. And we couldn't have done it without you. And I think all of you that have been involved with Amber really do appreciate who she is and how she's handled things. So thank you. So let's give her this is just a really special thing. And I know that all of you hopefully perceived it that way. We had just fabulous speakers, the students that we were able to have come here, other students, the interactions that I had, and I'm sure many of you had, make this really special for me. And I'm sure for Richie as well. And I just want to acknowledge the speakers and the audience. You guys, thank you all for traveling and for really, putting out your best efforts for us here at this meeting. It's just been terrific. And I also want to thank Richie as a partner for many, many years and a great friend. And having this event is fun because it sort of rekindles everything that we've got going together, and it's really quite nice. So.
Thank you, Ned. And I feel just the same, and it's really wonderful to keep doing this. I want to also express my appreciation to the students who participated in the spring seminar that is focused on the readings that you all gave us, and also to NIMH for supporting our emotion training grant. And Ned and I say this every year, but this was really the best symposium. So we're really so grateful. Thank you.